So it's Lispro, it's the same product, it's just formulated so that it's absorbed faster. You're on Humalog, aren't you?
I'm actually a Novolog right now, but I'm trying to think of the one that was absorbed fast. I think it was Keto, but they said.
Yeah.
We should all just sit really high. Some lab mentions another lab, so we're like, so I really want to be like in one of those labs because all the labs I've been so far, the lab is just so small for a tall person, and I'm just like, hi. Oh, this one, oh my god. That's bad. Yeah, this is tall, this one goes, but this one doesn't go down.
Aaron, where were you for 404?
Yeah, I didn't get a lot of sleep last night, and I took a nap and I worked on.
Okay. Are you going to get that dealt with? You're going to get that dealt with?
Yeah, I've got to sleep today over for Thanksgiving break.
Okay. Okay.
Yeah, I don't feel like I ever actually woke up this morning. I drove here and then wow, I made it to school.
Oh, one of those days.
Are you in a high chair or something? Or? Oh, okay. I was ready to put a bib on you and feed you or something.
I'm hungry.
Crystal tested negative. So she has 5 more days and then she's done.
No, I'm actually done Thursday. And then Friday, I should be able to come to class.
Friday she'll be able to come to. I thought you said in your email 5 more days.
Oh, I don't think I did, but I could have easily made a mistake and wrote, like, I don't know. But yeah, Thursday
Right.
at 8 p.m. is when I'm free.
Excellent. Excellent. Okay. Okay, it's 2:40. It's time to get started. I received an email from Katja's RD asking me to please excuse her from class today. She was very nonspecific, as well she should be, to respect Katja's privacy, but she said she received some extremely bad news and will not be able to come to class. So I think it's incumbent upon us to pray for her, saying whatever that bad news was, if it's bad enough for her not to be here, knowing the kind of person Katja is, how serious she is about her career and learning, is probably pretty bad. So let's keep her in mind, not just before class, but really for the whole time, because she is a friend of ours. We love her dearly, and we hope that whatever this is, it gets worked out. however, it's intended to work out. Let's start, therefore, with a prayer. Lord, we come, our hearts are somewhat heavy for Katya. We thank you. Most of all, Lord, we pray that you would be her comfort, that you would be her life, her light. Lord, that your body that you sacrifice would truly be real drink for her. Your blood shed for her would truly be real drink. And that just as those who partake of you in that way live in you and you in them, may you live in her. May you give her true hope and true joy, regardless of what the situation is. Bring her through this, please. Bring her back to us safely, soundly. Thank you for today, that it's warm. And now help us to go about the work of learning about glycolysis. In Jesus' name, amen. So before glycolysis, we're going to revisit our carriers for oxidation reduction reactions. We looked at the nicotinamide adenine dinucleotide carriers, and they have this structure, and that glare off the bench there is blinding. I may have to stand over here. Can that close? Can that be closed? Yeah, close that, please. It is maddening. Oh, yeah, it's got to go over the. Okay, well, Meredith is risking her life for us. So these, again, they're built upon the backbone of ADP, and then you also have an added ribose, and then a nicotinamide group. Remember, the nicotinamide group comes from dietary niacin. Dietary niacin. If you lack dietary niacin, you'll have trouble with oxidation reduction reactions, and you will have pellagra. Pellagra. If you ever read the incredibly depressing novels of John Steinbeck, and yes, they are depressing. He's a great writer, but depressing nevertheless. You'll read about pellagra in books like The Grapes of Wrath, where people are starving. His novels are very much an apologetic for Roosevelt's New Deal policies during the Depression. And pellagra is mentioned, and in dogs, it's called black tongue because the tongue actually turns black. And pellagra is characterized by the 3 Ds: dermatitis, diarrhea, and dementia. None of which sound very good. So that's pellagra. And you need it because it being niacin, because you need NAD and NADP. So you get this substantial change in absorption at 340 nanometers when it's reduced. That is used universally in biochemistry labs for enzyme assays that use NAD or NADP. When it's reduced, you can track the appearance of this. And because these enzymes are often markers for certain organelles, you could use them as assays for the purification and to follow these organelles as you purify them. So there's lots of enzymes that use these, well over 200. They're found throughout biology, bacteria and barn animals and fish, ferns, people named Fred, so on and so forth. They're your mother-in-law, Frida, and they're called most commonly dehydrogenases, less commonly oxidoreductases. And the nicotinamide ring, remember, is planar. You will accept the electron, pair of electrons as a hydride, You go from this aromatic group now to more of this thing with the 2 double bonds here. Whether or not the accepted electron is above, or excuse me, hydrogen is above or below the ring is going to be specific to the enzyme. Remember, this is carried as a hydride. The other hydrogen ion goes into solution. That's the same for NADP as well. So that stereospecificity carries over not just for ones that use NADP, but also NAD. And you can see the specificity here. I won't read you the riot act on these, but you can see that if they are stereospecific, they will create NADH exclusively in the A manner or the B manner or whatever. No, not necessarily. Now, flavines. Flavines carry 2 electrons and 2 hydrogen ions. They are also found throughout biological systems. So they have the, once again, ADP, there we are, then another ribose. I'm seeing a theme here. Then you have the isoalloxane ring. The isoalloxane ring is derived from dietary riboflavin. Dietary riboflavin. This comes in the form of FAD, flaving adenine dinucleotide, or FMN, flaving mononucleotide. So this whole thing is flaving adenine dinucleotide. See the adenosine, flaving adenosine dinucleotide. This FMN, flaving mononucleotide, just has this piece here, not this. Now, flavines have a versatility NAD does not have. So the reduction happens with the isoalloxazine ring, and I can show you that here. You will add your electrons, protons, and you get the reduction essentially here and here. So if we draw essentially this piece here with our marker here like that, maybe we could do that if we use our little pen tool. Use a highlighter. And so let's highlight here to here to here. That's a piece that's going to do the accepting right there. And we have the addition of 1 hydrogen ion and electron, and then a second for the completion there. So this first gets reduced to a semiquinone, and then the semiquinone to the fully reduced version. There's your isoalloxazine ring right there. That's the business end. Yeah.
So what does flavin have different from NADP?
I'm going to get to that here in just a bit. Now, NAD and NADP usually are not covalently linked to the enzyme. There are exceptions, but usually it's soluble. So the delta E of NAD stays the same. Flavines tend to be covalently bound to the polypeptide chain. That microenvironment in which it finds itself changes the delta E of the flavine. That increases their versatility when it comes to reactions. So whereas NAD has a delta E of minus 0.32 volts, or 320 minus 320 millivolts, whichever scale you want to use, the delta E of flavines is going to vary from 0 to minus 0.219. So basically 0 to minus 2.2, 0.22. Here are some enzymes that have flavines covalently attached to them, and these are some of the ones we will use and talk about in the course of these lectures. Some have FED, some have FMN, so on and so forth. Jenna? Well, structurally, it's just this piece. So if you just have the phosphate, that makes it less bulky. If you have this and you're bound to an enzyme, you have to have a nucleotide binding pocket to bind this. If you have this, you don't. So that's the issue of this. If you want this to be sticking out somewhere, so it has exposure to whatever your substrate is, that's your man or woman or whatever. If you're bound to a Rossmann fold, then that's your one there. So it depends on how the protein accommodates it. Both of them can accept both protons and electrons. Both of them vary according to how they're bound to the protein, what kind of protein they're bound to, microenvironments they're bound to, complexes they're in, that sort of thing. They both do the same job. All right, that is essentially bioenergetics. There's just one thing I'd like us to consider. So potassium ions, let's consider potassium ions. If you measure potassium ion concentrations in muscle, they're 124 millimolar in muscle. That is 60 times the potassium concentration that you have in serum. What about for sodium ions? Sodium ion concentrations in muscle are 4 millimolar. But in serum, it's about 140 millimolar. Now, those are kind of gee whiz numbers. The thing I want you to appreciate is the amount of energy it takes to do this. This takes a good deal of energy. So this is maintained by the sodium potassium ATPase, but let's consider a situation that's a little simpler because sodium and potassium, they're charged. Let's start out with something that's not charged. So let's start out with everybody's favorite molecule since Halloween was here, glucose. Now, let's say you want to take glucose and put it into a cell. And let's say the glucose concentration, so here's a membrane, here's the glucose. You want to put it into the cell. So let's say, or, oh, maybe you want to excrete it from the cell. And let's say, so, okay, let's do this. Liver cell, you just broke down your glycogen. So Ram goes on his, what are you doing, another 5, or are you doing, you'll do 5 today? He's doing another 5 today. And he didn't eat a very good lunch, or maybe he forgot to eat lunch. Okay, in which case it would be grounds to chew you out. And now he has to work on his glycogen in the liver. So let's say the concentration difference is 1,000 times. So you're working against a 1,000-fold concentration difference. What kind of energy would it take to pump this glucose from here to here? You're pumping it against concentration gradient of 10 to the 3rd. Let's calculate that just to give you an idea of the kind of energy requirements cells may have. So we're going to look at change in, this is the change in potential energy. So it's going to be the, oh, and this is at 37 degrees C. So how many Kelvin is that? Did I do that right? Good. Okay, good. I'm not saying things. Okay. All right. So we've got the gas constant, temperature, which that's what this will be. And this is going to be, oh gosh, I looked this up. It's 8.314 joules per mole Kelvin. This 310. That's 310 Kelvin. That's gonna, okay, right? Then we're gonna multiply that by natural log of our concentration, which would be 1/1000. And if I punch this up properly on my calculator, should be something like 17.8 kilojoules per mole. Not very favorable. Not very favorable. So if the glucose concentration is high, so let's say Greg goes trick-or-treating. Let's say Greg dresses up as Batman, okay? Greg goes as Batman and his costume is really cool, so he gets rewarded big time. So he gets, oh, Reese's Pieces, M&Ms. You're allergic to nuts? Okay. Okay. Well, plain M&Ms then. Okay. M&Ms, Kit Kats. What else would you have that's good? Starburst, Skittles. Oh, okay. That sort of thing. And Greg decides he worked hard. He deserves to reward himself, so he sits down and he has, oh, say about a quarter of his bag. And he gets a bit of a sugar high. All right, so now when the glucose goes into his cells, that glucose will be phosphorylated, as we'll see with glycolysis. That is going to trap the glucose in the cell. Therefore, its diffusion out of the cell will become thermodynamically unfavorable. However, that means that the glucose inside the cell, from a concentration perspective, isn't going to be glucose anymore because it's going to get metabolized very quickly. Okay, so that's the case if you're pumping against a gradient. That's not very favorable. Yeah.
You need a negative because if you do that, it comes out as
There.
minus.
Yeah, minor signs and whatever. Yeah. Okay.
It's a negative RT.
Yeah, it's a negative RT. It's a negative RT. Okay, let's take a physiological example here. Let's do chloride in your blood. Chloride in your blood. Okay, so chloride. Oh, good golly. Let's see. I can do this two-handed. All right. Now, I had a plant biochem professor named Harold Kahn. He'd write with one hand and erase with the other. So he'd do this, the whole class. Oh, you couldn't. You couldn't.
Look, I already do it.
It's, his grad student. I was sitting next to his grad student. She kept leaning over, telling me what he just written, because she knew what he was going to say, because she TA'd the class 3-4 times. She was leaning over to me, feeding me what was saying. So I had the best notes in the class. So.
And it's not even like they recorded lectures.
No, yeah, I helped a lot of people actually, but everyone said she was hitting on me. I didn't think so, but okay, chloride. I just think she wanted to show off how smart she was. Okay, chloride in the blood. So BL blood is about 100 millimolar. If we want to pump chloride into the urine, so you're a kidney cell, you want to pump it into the urine, you're pumping it into 160 millimolar concentration. Okay, let's calculate the work thereof, shall we? All right, so let's see, it's
So we want chloride out of the blood into the urine.
we want to go from, right, exactly. So if we want to pump chloride into the urine, we're pumping against concentration gradient. That requires work. Let's take a look at how much work this will require. So RT ln essentially in out. That's the same equation we're going to use. And now we're going to use a different value of R because this is just a smaller number. So we're going to use R is equal to 1.92872 calories per mole per Kelvin. Our K is equal to 310 again. Kelvin, so Kelvin, that cancels. And then, so we have, let's see, 1.92872 times 310. And then we have the natural log, well, let's see, natural log of 160 divided by 100. And if I got everything punched up correctly, that should be 290 calories per mole. Yes? I'm trying to save all those darn decimal points, so that's why I did it in calories this time. Because 0 point, so on and so forth. So, yeah, I wanted a bigger number, and you'll see why in just a bit. Now, can we use this number to calculate how much ATP is going to be hydrolyzed through the transport of these chloride ions? Can we, to transport a mole of chloride ions, can we figure out how much ATP we're going to have to burn? Sure. So to do that, we take the, so free energy change. Just take the free energy change of ATP hydrolysis. And then divide that by the energy required to transport the chloride ions. And that'll give us number of ATP to transport a mole, right? And if you punch this up, so it should be 10,000 calories per mole. And we're going to divide that by our 290 calories per mole. 290. And that should give me, I think, I came out to 34. That makes some mathematical sense. Yeah. Now, we have a problem here. Anyone know what the problem is? Chloride's charged. Chloride's charged, not like glucose. So this equation over here, we have to add an electrogenic factor to it, don't we? That's probably why this number seems kind of high. Well, if we're going to add an electrogenic factor, that electrogenic factor is going to be, that's the charge on our moiety, our old buddy Faraday's constant, and then delta V. That's our electrogenic factor that we're going to have to add to this. Now, delta V is going to vary from cell to cell. Depends on the cell type. It could be anywhere from varies quite a bit. So if we use a delta V that is more logical for a kidney cell, our delta V is going to be 120. That's going to be the voltage drop across the membrane. Okay. If that's the case, then we should be able to put all this in there, right? All right. Well, if that's the case, this is a monovalent ion, right? So that means this is 1. And our temperature again is 310. Delta V is 120. Delta V, you stick an electrode in the cell and you measure it.
Okay.
You look it up.
See, it has to be given to us, or we.
Yeah, it has to be given to you.
So if we were at the.
Yeah. Oh, yeah, all that information be given to you. Yeah.
Are you going to give us anything?
Yeah. But think about it, guys. If I gave you a lot of that info, you should be able to calculate the ratio of chloride in a case like that, couldn't you? Because that's a constant. Chlorides, you know what that is. That's the number I give you. That's a constant. That's a constant. You solve for this, so you could potentially discover this if you wanted to, right? Sure could. So these are things essentially, huh? Yeah, from that number, you could essentially solve for that if you wanted to. So this essentially is how you could use this math essentially to get something that it's actually metabolically pretty relevant for what you're looking at. That's the reason I bring this up.
I thought that wasn't accurate because we haven't added.
It's not, so we'd have to redo it. And if you did redo it, I won't take the time now to redo it. I'll leave that to you.
So over there with that math, what were you looking for?
Looking for how much ATP it would take to pump a mole of chloride into the urine. So how much work is it going to take essentially to? Okay, enough about bioenergetics. Let's talk about something else now. Namely, let's talk about glycolysis.
Does that sound right? About 3 ATP molecules?
Yeah. All right. Back to the ranch here. So when a cell takes up glucose, glucose is subjected to a step-by-step degradation. This step-by-step degradation is a biochemical pathway that is absolutely universal. It is known collectively that those steps collectively are called glycolysis. The pathway gets called lots of things, but it's named after the people who discovered it and characterized it. So it's usually called the Ebden-Meyerhof-Parnas pathway. And so some people call it the EMP pathway, or you can just call it glycolysis. So glycos in modern Greek means actually dessert. Glycos is the word from which we get glucose. Lysis comes from lysis, which means to destroy. So this is the glucose destroying pathway. The reactions of glycolysis are universal. You can take enzymes from bacteria and express them in insects. They still work. You could take a human glycolytic enzyme, express it in a bacterium. It still works. The pathway is that universal. Because there are few organisms that use a variation on it called the Entner-Duderoff pathway. So it is virtually universal. They just do. They just do.
What was the pathway in the other organisms called?
Entner-Duderoff, which we'll get to after we talk about glycolysis. So glycolysis has two parts. There is a first part where you prime the pump. You prime the pump, and by priming the pump, I mean you take glucose, you put phosphates on it, you make it this really high energy molecule. So breaking it down will yield ATP. Then you take that high energy thing, you break it down, make ATP. That's your payoff phase. Your payoff phase. So the preparatory phase. You take glucose, phosphorylate it, rearrange it, phosphorylate it again, then break it in half. And you essentially convert it to 2 molecules of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate will then go through the steps of the payoff phase. This is glyceraldehyde. So here, there's essentially glycerol, except that alcohol has been oxidized to an aldehyde. So it's glyceraldehyde. You start from here, 1, 2, 3, glyceraldehyde 3-phosphate. And these are the steps which we will cover in gory detail, and I do mean gory. And you can see there's 4 steps. The preparatory phase requires the expenditure of 2 molecules of ATP. And you get from this 2 molecules of glyceraldehyde 3-phosphate. The payoff phase oxidizes glyceraldehyde 3-phosphate to pyruvate. And in the payoff phase, we get 2 ATP per glyceraldehyde 3-phosphate. So we get a total of 4, which gives us a net gain of 2 ATP per glucose. 2 ATP per glucose. You also generate 2 reduced NAD molecules. So you get 2 NADH per glucose. And here is the payoff phase, steps 5 to 10. So 1 to 4, preparatory phase. 5 to 10, payoff phase. Preparatory phase, priming the pump. Payoff phase, you're oxidizing glyceraldehyde 3-phosphate to pyruvate. Amanda?
So it's 1 glucose and 2 ATP then, and then out is 4 ATP and 2 NADH.
Yeah, and 2 pyruvates.
Okay.
So the logic, if you will, of glycolysis essentially is shown here. And this logic of glycolysis will make more sense when we go through the individual steps. You learn the individual steps and you look back on it. But essentially, each of these has a chemical logic to it. And the chemical logic involves generating 1,3-bisphosphoglycerate, high energy molecule, and phosphoenol pyruvate, another high energy molecule that'll be used to make an ATP, that'll be used to make an ATP. That's the logic, if you will. And we'll get to the logic once we cover the individual steps. You need to know the structure of every intermediate. You need to know the name of the enzyme. And if we go over the mechanism that the enzyme uses, you have to know that too. So you have to know the names and structures of the intermediates, the enzymes involved that catalyze the reaction, and any mechanisms that we pause to mention. So we usually start with glucose. We will see you can also start with other sugars, but most of the time we're starting with glucose. First step, you're going to phosphorylate glucose. Now the logic of this is that a phosphorylated glucose is trapped inside the cell. It's now committed to glycolysis. It can't leak out of the cell. So you're going to use ATP. You'll phosphorylate glucose on carbon number 6. You make glucose 6-phosphate. And the enzyme that catalyzes this is hexokinase. Hexokinase. Hexokinase is the most well-studied example of induced fit. When it binds its substrates, ATP and glucose, it undergoes a remarkable conformational change. The enzyme essentially is like this. It binds its substrate and it kind of bites down on the substrates like that. And that essentially generates the catalysis of the reaction. And there's a picture we can look at. And essentially, oh boy, I used to have a video that showed, okay, induced fit movie. It's not showing the induced fit movie. Okay, back to the ranch here. So this essentially is kind of what happens with the induced, oh, that's so glary. So the enzyme kind of has a mandible and a maxilla, if you will. And then when it binds the substrate, see how it kind of bites down on the substrates? And it binds the two substrates pretty much simultaneously. And the binding of those will take the two substrates essentially and put them in a proximity so that the reaction occurs very, very favorably. And you get from this ADP and glucose 6-phosphate. ADP and glucose 6-phosphate. Then it releases the substrate, goes back to its original conformation. Hexokinase comes in different types of isozymes. What's an isozyme? Cells sometimes have proteins that are either encoded by the same or different genes, but they have different amino acid sequences. How different varies. These proteins are different enough that you notice them, but they catalyze the same reaction. So these are called isozymes. Hexokinase comes in 4 different isozymes. They're given really creative names. Want to hear this? Hexokinase A, hexokinase B, hexokinase C, and you guessed it, hexokinase D, hexokinase D. Now, hexokinase isozymes A, B, and C can phosphorylate glucose, mannose, or fructose. They can phosphorylate glucose, mannose, or fructose. Hexokinase A, B, and C are encoded by different genes that are located on different chromosomes. Hexokinase 1 is found almost in every tissue. Hexokinase 2 is restricted to insulin-sensitive tissues like fat, skeletal muscle, and cardiac muscle. Yeah, the genes are called hexokinase gene 1, hexokinase gene 2, hexokinase, which is confusing as heck. But the proteins are called hexokinase A, hexokinase B. The molecular biologists drive me nuts. Call them something cool like blue unicorn or snake or catastrophic, whatever. Yeah, whatever. Genes. They're on different chromosomes. Yeah, everywhere. Just about almost every mammalian tissue has hexokinase A, almost. B is limited to insulin-sensitive tissues, skeletal muscle, fat, cardiac muscle. Interestingly enough, hexokinase B is expressed at very high levels in cancer cells. And when cells switch to high levels of hexokinase B, that's correlated with poor survival rates. Nobody knows why. That's a tidbit of worthless information. Hexokinase C is kind of everywhere at low levels. And I will right now. So hexokinase D is also called galactokinase. Galactokinase. This is, yeah.
Sorry, is it galactokinase or glucokinase?
Sorry. Galactokinase is for galactose. Glucokinase. Glucokinase. So this is restricted to hepatocytes, that is liver cells, and pancreatic beta cells. There's also low expression in the hypothalamus. Nobody knows why. Might have to do with regulating appetite. This enzyme has a very high Km for glucose. So put on your enzymatic glasses. What does that mean? High Km for glucose. You need lots of glucose to be able to saturate the darn thing. So when are you going to have lots of glucose in your blood? After you go on that post-Halloween sugar binge, right? Or something like that. And this enzyme is the sensor in your liver for high amounts of glucose in the blood and in the pancreas as well. So here is the reaction. Students often ask me, how did I learn these? I learned them by drawing them out constantly. During the old days, the computers had those doily wheel printers. So there was tons of extra paper at the campus computing center. I would go and steal their extra paper, and I'd sit there and I'd draw them out and draw them out and draw them out. If I could draw them out 4 times without looking at the book and not making a mistake, I figured I learned it. And then I went on to the next one. And after I learned the next one, I tried to draw both of them 4 times without making a mistake. That's just what it took for me. Yeah.
There's at my internship, one of the girls found out I was in biochemistry and actually told me to help remember the steps of glycolysis. There is a parody to Macklemore script shop that's really helpful in memorizing every step. I don't know it yet, but trust me, I will.
Yeah. Guys, notice here. See the cofactor? What did we say about divalent cations in ATP? They shield the negative charges, and if enzymes use ATP, they will very commonly employ a divalent cation like magnesium. So I get glucose on carbon number 6. Carbon number 6. Notice it's drawn in the Pyrenees ring, because that's the predominant ring in the aqueous form. And it can be alpha or beta, it doesn't matter. Just know this negative. Yeah. Second step. Now, glucose, you're going to rearrange it. You're going to rearrange it to fructose. You have to, because the next enzyme only uses fructose as a substrate. So you're going to rearrange this to fructose 6-phosphate. Fructose 6-phosphate. And this is an isomerase that does this. This is phosphohexoisomerase. Phosphohexoisomerase. This uses magnesium ions as a substrate, as a cofactor. So you convert glucose 6-phosphate to fructose 6-phosphate. And now, see that hydroxyl group there? It's just sitting there waiting to receive another phosphate, isn't it? That's why you convert it to fructose 6-phosphate. The mechanism of this enzyme is very interesting. So this particular ring can undergo opening and closing, opening and closing, opening and closing. When it opens, the enzyme grabs it. There's a basic group here. That basic group will abstract this hydrogen. When it abstracts the hydrogen, you make a double bond. You think a rearrangement of bonds here. This leaves. Then the hydrogen it abstracted, it gives it back to make the alcohol. That's how you go from aldehyde to alcohol. Take the proton off, give it back. That easy. The basic group will vary from enzyme to enzyme, but virtually all enzymes use this mechanism of abstracting, giving it back. Abstracting it, letting it rearrange, giving it back.
So we should know the sign.
Yes.
Okay, cool.
Yeah.
I got it.
So you have a cis enediol intermediate. That's pretty universal for phosphohexoisomerases. Next step, you're going to burn another ATP. You're going to use ATP. You're going to convert fructose 6-phosphate to fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. This is the enzyme phosphofructokinase. This is a highly, highly, highly regulated step of glycolysis. This is the no turning back step of glycolysis too. Once again, look at the cofactor. Yep. Now, it's called PFK1. There's another PFK. It's a regulatory enzyme that's called PFK2. We'll see what it does in a bit. But this is a glycolytic enzyme PFK1. PFK1. This delta G is also very, very negative. This delta G, not so negative. How do you pull this through? By getting rid of that product very quickly. So as soon as you make that product, PFK jumps on it and boom. That's how you pull this through. This is an allosteric enzyme. Again, very highly regulated. So it is a dimeric enzyme. Dimeric enzyme. Notice that your active sites are here. The effector sites, where the regulators bind, tend to bind at the interface between the subunits. That's quite important. And remember, your substrates are fructose 6-phosphate and ATP. Fructose 6-phosphate and ATP. This enzyme has a Rossmann fold. Yeah, it has a Rossmann fold. That's where the ATP binds. So here is your fructose 6-phosphate. Here's the hydroxyl group. Here's ATP. Here is this poor, lonely, abused phosphate. All his electrons being taken by these mean old horrible oxygens. Here is the electron-greedy oxygen spying that slightly positive phosphate. And what does the greedy old oxygen do? It attacks the poor abused phosphate, henpecking it almost to death so that the bond here is broken. It abstracts it away, and you get ADP. And now this phosphate goes down to there. Fructose, 1,6, bisphosphate. Sorry, that should have 2. That has 3. Yeah, just scratch that out. Okay, notice stabilization by magnesium. That's very important. Very important. PFK2. PFK2 makes a molecule called fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is an allosteric regulator of PFK1. So PFK2 takes fructose 6-phosphate and ATP and makes a different molecule, fructose 2,6-bisphosphate. If you have a decrease in ATP concentrations, PFK activity goes up. PFK activity goes up. If you have an increase in ADP or AMP, PFK1 activity goes up. If you have a large supply of fatty acids, that's an alternative energy source. PFK activity goes down, tends to inhibit PFK1. Fructose 2,6-bisphosphate is a potent allosteric activator of PFK1. Very, very potent.
So it's working in the reverse when you have a decrease of ATP in the cell. It's catalyzing the
Yeah, it's because it's an ATP, part of an ATP making pathway. So make more ATP. Now, if you have too much ATP in the cell, that actually inhibits PFK1. Step 4 of glycolysis. You're going to take fructose 2,6-bisphosphate and you're going to do an aldol condensation in reverse. So this enzyme is called aldolase. Aldolase. And you will convert it to 2 3-carbon phospho, 2 3-carbon compounds that have phosphates on them, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate.
You do both of those every time.
Yep,
You get one.
yep. And then there's an isomerase that converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Sorry, 1,6, 1,6, 1,6, 1,6, 1,6, 1,6, 1,6, just like shown here. Now, this ain't so favorable, is it? So that glyceraldehyde 3-phosphate gets metabolized really fast. That way you pull this reaction through. The mechanism of this enzyme is really interesting. It uses a Schiff base intermediate. That's interesting, but we don't have time to talk about it. So remember your quiz Friday is on bioenergetics. We'll cover the rest of glycolysis on Friday.
So glycolysis is not going to be on the list.
No,
So on the syllabus are April due next week Friday. Maybe give me that one.
when I get it written,
The mood.
which won't be this weekend. I've got a quiz this weekend, so I won't have any time to write it.
So it'll give you like a week when we get it.
Yeah, a week from when I get it to you.
Okay, cool. I have more things to do.
Okay, guys. That's it for today. See ya.